some of the previous videos, we have discussed in detail about the various methods of pollination in flowering plants. In this video, let's discuss in detail about the process of fertilization in flowering plants or angiosperms. So in flowering plants, this entire process of fertilization was actually discovered by a botanist whose name was Strasburger. And as we have learned this in lower grades, we know that in the process of fertilization, the male gamete fuses with female gamete and produces a single-celled diploid structure which is called zygote. And this process of fertilization is also known as syngamy. So the syngamy of fertilization in flowering plants was discovered by Strasburger. We know that in case of flowering plants, the male gamete is pollen grain, or you can say the male gamete is actually present inside the pollen grain, and female gamete is egg cell or ovum. Okay, and this egg cell or ovum is actually present inside the ovary, right? Now, the first very important step in the process of fertilization is pollen-pistil interaction. Pistil is the female reproductive organ of the flowering plants. Now here comes the significance of the process of pollination, in which the pollen grain falls from the anther to the stigma or on the pistil of another flower, or maybe of the same flower. Okay. So during the process of pollination, what happens is the pollen grain falls, or pollen grains are transferred from anther to the stigma of the same flower or maybe of different flower. Okay. Now once this pollen grain actually falls on the stigma of the pistil, okay, pistil is having three parts: stigma, style and ovary. So once this pollen grain falls on the stigma of the flower, or you can say stigma of the pistil, so first this stigma actually tries to recognize this particular pollen grain. Okay, you can say stigma or pistil tries to recognize this particular pollen grain. Okay, it tries to recognize whether the pollen grain is compatible or incompatible. Okay, now the meaning of term compatible is this pistil actually tries to identify that the pollen grain which has actually fallen on it belongs to the same species or it belongs to the different species. For example, there is a pollen grain of marigold flower, and this pollen grain of marigold flower has actually fallen on the on the stigma of rose flower, or maybe on the stigma of hibiscus flower. So what will happen here? Now the pollen grain becomes incompatible because the pollen grain belongs to the different species. Rose plant is of different species, and marigold plant is of different species, isn't it? But if the pollen grain of marigold flower falls on the stigma of the another marigold flower, okay, so now here the pollen grain becomes compatible because both these marigold flowers, they belong to the same species. Okay, and this is what happens in pollen pistil interaction. During pollen pistil interaction, the pistil actually produces a sugary liquid. Okay, it actually produces water and sugar, and which is called sugary liquid, and certain chemicals which actually helps the stigma in recognizing the pollen grain, whether it belongs to the same species or it belongs to the different species. Okay, and if the pollen grain is compatible, that means if the pollen grain is of right type, if the pollen grain belongs to the same species, then pistil will accept it and it will promote post-pollination events in the plant. Right now, the next very important step after pollen pistil interaction in the process of fertilization in flowering plants is germination of pollen grains. Okay, this is actually pollen grain. Okay, now this step is very important because once this pollen grain germinates, it produces pollen tube, and this pollen tube is the structure which is actually going to carry male gamete from stigma to the ovary of the ovary, or you can say from stigma to the female gametophyte, or the egg cell which is present inside the ovary. We have already studied about the structure of pollen grain. We know that this pollen grain, the outer side is covered with a very tough covering which is actually known as exine. Okay, this is a very tough covering which is called exine. And but this exine layer is actually not continuous. Okay, this is discontinuous. There are certain areas or certain points on the pollen grains where this exine is absent, and these areas are actually depressed in the center. Okay, and these areas are known as germ pore. Okay, so there are multiple germ pores present on every pollen grain. Okay, now what happens is during this particular step, the pollen grain which has actually fallen on the stigma, and the pollen grain is of right type. So this pollen grain germinates, and through this germ pore, the pollen tube forms. Okay, the pollen tube grows out of this germ pore. Okay, now there are certain species in which during the germination process only one pollen tube is formed. So such type of species are known as monosiphonous, or you can say monosiphonous is the term which is actually used to describe the formation of only one pollen tube through one germ pore in the pollen grains of certain species. Okay, but there are certain species of angiosperms in which more than two pollen tubes are formed through the germ pores, and these species or maybe this process is known as polysiphonous. So here poly means many, and here mono means one. Okay, so monosiphonous is the condition in which only one pollen tube is formed through the germ pore of any pollen grain, and polysiphonous is the condition in which two or more than two pollen tubes are formed through the through two or more than two germ pores of the pollen grain. Okay, so in the process of germination of pollen grain, pollen tube is formed through the germ pore of the pollen grain. Now once this pollen tube is formed, now this pollen tube actually passes through the long neck like, or long filament like structure which is called style. Okay, now there are two types of style. In certain species, the style is hollow from inside. Okay, the style is hollow from inside, and one or two layer of cells are present only in the periphery. As you can see here in this diagram, the style is completely hollow from the center, and one or two layer of cells are present only in the periphery. Periphery means the corner of the style and the stigma. So in this case, the pollen tube can easily grow, and it can easily pass through the style and finally reach to the ovary. Okay, because inside ovary here you have this female gametophyte, and inside female gametophyte you have this egg cell. Okay, but there are certain species in which the style is actually not hollow. Okay, the style is completely filled with the cells. Okay, there are cells which are completely or tightly packed inside the style. So in such species, the pollen tube actually has to push these cells. Okay, push these cells and grow through the style. Okay, the pollen tube actually has to push these cells and grow through the style. Okay, so what happens is during the growth of pollen tube, as we have studied about the structure of pollen grain earlier in the previous videos, we know that pollen grain is covered with exine. Okay, it is also covered with intine. Intine is the inner layer and exine is the outer layer. Now this pollen grain is having two cells. One is tube cell. The larger cell is known as tube cell, and it is having its own nucleus and cytoplasm. And floating inside this tube cell is a spindle shaped cell which is known as generative cell. And this generative cell is the one which actually undergoes one mitotic division. Mitotic division means the division, the type of cell division in which like the equal number, the same number of chromosomes are passed on to both the daughter cells. Okay, so for example, if this generative cell is having 36 chromosomes, uh, okay, so in the male nucleus, the two male nucleus which will be produced, both male nucleus will carry 36 chromosomes each, 36 and 36. Okay, so mitotic division is also known as equational division, because in this the chromosome numbers remain same, okay, in the parent cell as well as in the daughter cells. So this was the entire structure of the tube of the pollen grain: exine, in time, then tube cell and generative cell. So during this growth of pollen tube, this when the pollen tube actually starts growing, so this entire content simply do not move inside the pollen tube. Okay, this pollen tube moves down by pushing the cells of the style, and during the growth, this generative cell, the small spindle-shaped cell which is floating in the cytoplasm of the tube cell, divides by one mitotic division, and it produces two male gametes. Okay, so this point is important. It produces two male gametes. Okay, and now this entire content, that means the cytoplasm of this pollen grain, the nucleus of the tube cell. Okay, remember this. What is the content? Cytoplasm of the pollen grain plus nucleus of tube cell plus these two male gametes. Okay, the male gametes which are actually produced after the mitotic division of generative cell. So all this entire content actually passes through the pollen tube. Okay, now this entire content is actually present present in the at the tip of the pollen tube, almost at the tip of the pollen tube. Okay, now when this pollen tube is actually passing through, it is actually pushing down the cells of the style, and it is passing through the style. So at that time it might happen like for example, if the pressure is generated here in this particular part, so what will happen? It might happen that half of this content, that means cytoplasm, nucleus of the tube cell and two male gametes, so half of this content might remain in the upper part of the pollen tube, and half of this content might go or remain in the lower part of the pollen tube. Okay, so in order to prevent this, what happens is callose plugs are formed at interval. Okay, this black color, the dark color, this dark black color structure you can see here, this is actually known as callose plug. Callose is actually a polysaccharide. So this callose plugs are formed in the pollen tube, and the significance of formation of these callose plug is, once callose plug is formed here, and now the lumen is actually completely sealed. Okay, and the entire content of this pollen tube is present here. So when this pollen tube is trying to grow downward, so this content of the pollen tube will not go back. Okay, it will not go back in the upward direction. Okay, so this entire content of the pollen tube, that is cytoplasm, nucleus of tube cell and two male gametes, they all should remain together in this entire growth of the pollen tube. And how they remain together? Because callose plugs are formed at intervals in this pollen tube. Okay, if this is a pollen tube, so first the callose plug is formed here, and when this content moves ahead, the next callose plug will be formed, and then again the next callose plug will be formed as the content of this pollen tube, that is cytoplasm, nucleus of the tube cell and two male gametes, when they are moving downwards in the pollen tube. Once this pollen tube completely crosses the style and reaches the ovary, now this pollen tube actually has to enter the female gametophyte. Okay, we have already discussed the structure of female gametophyte, or you can say embryo sac, in one of the previous videos, and there you have learned that this female gametophyte is having two ends. Chalazal end, okay, this end is known as chalaza, and this end is known as micropylar end. Okay, so this particular this particular pollen tube has to enter inside this female gametophyte. Okay, in order to reach the egg cell and perform the process of fertilization. So there are three ways in which the pollen tube enters inside the female gametophyte or the embryo sac. If the pollen tube enters inside the female gametophyte through chalazal end, okay, as you can see here in the second picture, this particular female gametophyte is actually particular female gametophyte is actually receiving pollen tube through the chalazal end. You can see here pollen tube has actually entered inside the ovule through this chalazal end, and then it actually uh, through this chalazal end, you can see it through this chalazal end, and then it actually tries to fuse the egg cell. So this process is known as chalazogamy. Okay, so chalazogamy is the method in which pollen tube enters inside the female gametophyte or embryo sac through chalazal end. Second is second type of method is known as mesogamy. Okay, so mesogamy, here you can see the third image. In this you can see it's a method in which actually pollen tube enters inside the female gametophyte through integuments. Okay, you can see these are the two layers of integuments, outer integument and the inner integument. So pollen tube here is entering inside the female gametophyte through integuments. Okay, and the third and the most common method which is present generally in all the angiosperms is porogamy. Okay, poro refers to the pores. So porogamy. So porogamy is the most commonly available method in all angiosperms, and this is a method in which the pollen tube actually enters inside the female gametophyte through micropyle. Okay, it enters inside the female gametophyte through micropyle. So this is how pollen tubes enters inside the female gametophyte of angiosperms. There are three ways, either chalazogamy, mesogamy or porogamy. Now, once this particular pollen tube actually reaches the female gametophyte, so what happens is, we know that at the micropylar end, okay, at the micropylar end, this is the micropylar end and this is the chalazal end. On the top of chalazal end here we have three antipodal cells. Okay, in the center we also have like this one is known as polar nuclei. Okay, and at the micropylar end we have two synergids. So these green color cells are actually synergids, and this purple color cell is actually the ovum or the egg cell. This is the female gamete. Okay, now when this pollen tube reaches the female gametophyte through this micropylar end, so what happens is, one of the synergid out of two, one synergid actually degenerates. Okay, one of the synergid degenerates, and each of the synergid is actually having a filiform apparatus. Okay, this is having the filiform apparatus. So one of the two synergid actually degenerates, and this filiform apparatus of the degenerating synergid guides this pollen tube to enter inside the female gametophyte. Okay, and when this pollen tube enters inside the female gametophyte, it will release its content into the female gametophyte. Okay, so what happens is, I've told you in the previous slide that in this pollen tube right now, the male two male gametes are present, the cytoplasm of the tube cell is present, and the nucleus of the tube cell is present. Okay, so the moment this pollen tube reaches here in the female gametophyte, the one of the synergid degenerates, and the filiform apparatus of that particular degenerating synergid guides this pollen tube to enter inside the female gametophyte. And during this process, the tube cell, okay, the tube cell nucleus, the nucleus of the tube cell degenerates. Okay, so the moment this pollen tube reaches the female gametophyte, the nucleus of the tube cell here will degenerate, and only the two male gametes will now enter inside the female gametophyte. So one of the male gamete goes and fuses with the egg cell. Okay, so one of the male gamete out of two goes and fuses with the egg cell. So this process is known as fertilization or syngamy. Okay, so it's a process of fusion of male and female gametes and the formation of single-celled diploid structure which is called zygote. Okay, so remember, for example, this is the female gametophyte. Here you have 3 antipodal cells, and here you have, like, one degenerating synergid, one normal synergid, and here you have the egg cell. And in the center you have polar nuclei. Okay, so what happens is, when this pollen tube enters here in the female gametophyte, so 2 male gametes are released into the female gametophyte. One male gamete fuses with the egg cell, okay, and it produces the single-celled diploid structure which is called zygote. Okay, and remember, the male gamete and the female gamete, they are both haploid. Okay, and together they fuse to produce the diploid single-celled zygote. Okay, so this process is known as fertilization. Now, what happens to this second male gamete? Okay, we have learned that 2 male gametes enter inside the female gametophyte. One male gamete fuses with the egg cell or the ovum, or you can say the female gametophyte, and it produces the single-celled diploid zygote. Okay, now what happens to this second male gamete? So the second male gamete undergoes a very important process which is called triple fusion. Okay, now we have seen that in this female gametophyte, 3 antipodal cells are present at the chalazal end. Okay, in center we have these 2 polar nuclei. Okay, these 2 polar nuclei are present in the center. Okay, and here we have a degenerating synergid, one normal synergid, and this is actually the egg cell. So one male gamete fuses with the egg cell, and the second male gamete goes and fuses with these 2 polar nuclei. Okay, so the second male gamete, okay, this is actually the second male gamete, and the second male gamete is haploid plus 2 polar nuclei. Okay, 2 polar nuclei. These 2 polar nuclei are also haploid. Okay, so the second male gamete fuses with the 2 polar nuclei and results in the formation of primary endosperm nucleus. Okay, so the second male gamete fuses with 2 polar nuclei present in the center of the female gametophyte and results in the formation of primary endosperm nucleus. Okay, and this primary endosperm nucleus is actually triploid. Okay, this is actually a triploid structure. Okay, a triploid means it is having 3 sets of half chromosome. Okay, 3 half sets of chromosome. Okay, because all these 3 structures, that is second male gamete, then 2 polar nuclei, all these are haploid, isn't it? Hence, it forms the primary endosperm nucleus, which is actually triploid in nature. Okay, it is having 3 half sets of chromosome, and this entire process is known as triple fusion. Okay, triple fusion, because during this process of triple fusion, the 3 haploid structures are fusing with each other to form the primary endosperm nucleus. For example, you have a particular plant, maybe corn plant. Okay, so if the male gamete of this corn plant is having 12 chromosomes, okay, the male gamete, listen carefully, the male gamete is having 12 chromosomes. So how many chromosomes will be present altogether? How many chromosomes will be present in the primary endosperm nucleus? Okay, the cells of primary endosperm nucleus. Okay, so we know that here it is having 12 chromosome, the male gamete. Okay, and obviously the 2 polar nuclei, they will also have 12 chromosomes each. So the cells of this primary endosperm nucleus will be having altogether 36 chromosomes. Okay, so this method is known as triple fusion. Now in this entire process, you have seen that 2 times the process of fertilization takes place. Okay, 2 male gametes actually enters inside the female gametophyte. One male gamete fuses with the egg cell of the female gametophyte, and the second male gamete fuses with the two polar nuclei present in the center of the female gametophyte. Okay. So first process or first method in which male gamete fuses with the egg cell of the female gametophyte, this process is called fertilization or syngamy, and this results in the formation of diploid zygote. Okay, single-celled diploid zygote. And the second male gamete travels to the center of the female gametophyte and it fuses with the two polar nuclei present in the center. Okay, and it results in the formation of triploid endosperm, or you can say primary endosperm nucleus. Okay, so as the process of fertilization is taking place inside female gametophyte twice, okay, two times the fertilization process is occurring, this is called double fertilization, right? And double fertilization results in the formation of zygote and it results in the formation of endosperm, diploid zygote and the triploid endosperm. Okay, and this method is called double fertilization. So this process of double fertilization in flowering plants was actually discovered by another botanist whose name was Navaschin. Okay, his name was Navaschin. Right? This was all about the entire process of fertilization in flowering plants. In next video, we will continue with the development of primary endosperm nucleus and the formation of embryo from zygote. Take care. Bye-bye.